Watson and Crick has told that DNA replicates semi-conservatively. DNA replicates semi-conservatively. DNA replication is semi-conservative means in a DNA molecule, during the replication, what happens is the two parental strands will separate. That is, the two template strands will separate, and the new strand will be synthesized complementary to the parental strands. As such, when you see each DNA molecule has got one parent strand and one new strand, not entirely new strands. They have one parent strand too. That's why they say DNA replicates semi-conservatively. And it is Meselson and Stahl that demonstrated that DNA replicates semi-conservatively by using E. coli, Escherichia coli bacteria. They cultured Escherichia coli bacteria in medium that contains ammonium chloride, heavy isotope N15, ammonium chloride heavy isotope. So E. coli was cultured for many generations in this ammonium chloride solution. And then when the density was determined in cesium chloride density gradient, it was found that the DNA of the E. coli was found heavy because both the strands were developed from N15. Next, what they did is they transferred the E. coli to ammonium chloride light isotope, that is N14. Now the E. coli started to get cultured in N14 medium. After 20 minutes of their generation time, the DNA was isolated and the density gradient was checked through cesium chloride. What was found is that each of the DNA, since they separate, and the new DNA strand is produced from the ammonium chloride N14. That's why the density was of intermediate level. So they were intermediate density. They continued the culturing of E. coli for the next 20 more minutes. So after 40 minutes, they found the E. coli DNA when they isolated and when they cultured, when they put it in cesium chloride density gradient, they found that these DNA strands will be separating and they were of light density. So there was a band of light density, DNA consisting of light density. There was also a band of DNA having intermediate density, that is hybrid DNAs. Hybrid or intermediate density. Now this can be possible only if the DNA replicates semi-conservatively. Like if this was the DNA that was grown in N15 medium, then it will be N15, N15. After many generations, the DNA of E. coli will have N15, N15. That means it will be heavy in density. Then when they are transferring it into ammonium chloride N14, then the two DNA strands, parental strands will separate and the new DNA will be synthesized or replicated from the N14 medium. So one strand will be N14 and the other strand will be N15. And that's how we get hybrid density or intermediate density. Then they're going to continue in the same N14 medium for one more generation, that is after 40 minutes. Then what happens is that these DNA which is already existing, their strands are separating. And the new DNA which is produced are from the existing medium, that is in N14. So they were already in ammonium chloride N14. So the new strands will be synthesized from the N14 medium. That's why in the cesium chloride density gradient, we get two strands. One is of lighter density with N14, N14, and the other is of intermediate density with N14, N15, which can be possible. Like this, we will get only if the DNA was replicating semi-conservative. Hence, proved that DNA replicates semi-conservative.